What if I told you there's a secret code, a blueprint that connects you to every single living thing on this planet? Seriously, from the tiniest bacterium to the biggest blue whale, it's all written in the exact same language. Today, we're going to crack that code together.
Yep, you guessed it. That incredible instruction manual is DNA. And we're about to see exactly how this tiny molecule pulls off the grand symphony of life.
All right, so if DNA is the blueprint, what's it actually made of? What are the letters? What kind of ink are we using? Let's break it down to its most basic parts.
Okay, so picture DNA as this massive, colossal chain with literally trillions of links. Each one of those links, which we call a nucleotide, is made of just 3 simple parts: a sugar, a phosphate, and a base. And it's that last part, the base, that holds all the information. This simple little trio is the repeating unit that builds the entire code of life.
Now, here's where the magic really happens. It all comes down to a super simple, absolutely unbreakable rule. There are 4 types of bases, A, T, C, and G, and they only pair up in one specific way. Adenosine, or A, always hooks up with thymine, T, and cytosine, C, always pairs with guanine, G. This isn't just a suggestion, you guys. It's the fundamental law that makes the whole genetic language work.
So how in the world did we figure all this stuff out? Well, the first huge piece of the puzzle came from 2 scientists, Rosalind Franklin and Maurice Wilkins. By basically shooting X-rays at DNA, they got this fuzzy picture that revealed its shape wasn't just a straight line, it was a spiral, a helix.
And that X-ray image, it was the key. Building on that crucial piece of evidence, James Watson and Francis Crick were finally able to solve the puzzle. They built the very first 3D model of DNA, showing the world what the code of life actually looked like.
And this is what they found, the iconic double helix. The best way to picture it is as a twisted ladder. The 2 long sides of the ladder are made of those sugar and phosphate parts we talked about. And the rungs you'd step on? Those are the base pairs, the A's connected to the T's and the C's connected to the G's, holding the 2 strands perfectly together.
Okay, so we have this massive blueprint. But when a cell needs to divide, it has to make a perfect copy of billions of letters of code and do it flawlessly. I mean, how on earth is that even possible?
Well, the process has this really long name, semi-conservative replication. But the idea behind it is actually beautiful and honestly kind of genius. It just means that when DNA copies itself, each new molecule is half old and half new. It uses the original strand as a perfect template for the new one. That's the secret to making a perfect copy every single time.
It all happens in 2 main steps. First, a tiny little biological machine, an enzyme, comes along and acts just like a zipper, unzipping the double helix right down the middle. Then another amazing enzyme called DNA polymerase gets to work. It zips along each of the original strands and builds a brand new, perfect partner for it, strictly following that A with T, C with G pairing rule.
But wait a minute. If you were trying to copy a book with 3 billion letters, you're bound to make some mistakes, right? I mean, I know I would. So how does a cell handle typos in its own genetic code?
Well, the cell has this absolutely incredible quality control system. First, it literally proofreads its work as it's copying. Then, another system for mismatch repair double-checks the new DNA strand for any mistakes that might have slipped through. And on top of all that, there's even an excision repair crew on standby to fix damage from things like UV light from the sun. This whole system is so effective that it makes less than 1 error for every billion bases it copies. That level of precision is just mind-blowing.
So we can copy the entire library. Great. But what about reading just one specific recipe, one single gene, to build something the cell needs, like right now?
Here's the challenge. That DNA master plan is way too valuable to just leave lying around. It's locked away for safekeeping inside the cell's nucleus, like treasure in a vault. So to get the instructions out to the cell's little factories, it makes a temporary, disposable copy called RNA. It's a bit different, it's single-stranded, and it's allowed to leave the nucleus, which makes it the perfect messenger molecule. And it's not just one type of RNA. It's a whole team of RNA heroes working together. You have messenger RNA, or mRNA, that's the literal photocopy of the gene. Then there's transfer RNA, or tRNA, which acts like a fleet of tiny delivery trucks, bringing in the exact building materials needed. And finally, ribosomal RNA, or rRNA, is actually a part of the factory itself. It makes up the workbench where the protein gets built.
This process of making the mRNA copy from a DNA gene is called transcription. So the really important thing to remember here is we aren't changing the original blueprint at all. We're just making a quick, working copy of one small part of it.
And that brings us to the grand finale, the final spectacular act. How does the cell actually read that little RNA message and turn it into the real machinery of life, a protein?
The cell uses something called the genetic code to read the message. And here's the trick. It doesn't read one letter at a time. It reads the message in 3-letter words called codons. Each codon is a specific instruction to add one particular amino acid. And amino acids are just the small molecular building blocks that proteins are made of.
This whole thing happens at the ribosome in a process called translation. It starts by clamping onto the mRNA message. Then it just chugs along, reading each 3-letter codon one by one. For every codon it reads, those tRNA delivery trucks bring in the exact right amino acid, and the ribosome links them all together into a growing chain. It keeps doing this until it hits a stop codon, and then it just lets go of the brand new, fully formed protein ready to go to work.
Okay, so we've gone from the structure of DNA to copying it, to transcribing it, to translating it into an actual protein. When you boil all of that down, what's the core principle that powers all of biology?
It's this simple, elegant, powerful flow of information. The master blueprint is DNA. A working copy is made as RNA, and that copy is read to build a protein. This core idea, from blueprint to message to machine, is how a silent, static code explodes into the dynamic, beautiful, and complex action that we call life.